Hello, everybody, and welcome to this evening's program. We're quite impressed how many people actually have joined. We see over 200 people already on. So in a minute, I'm going to introduce Dr. Tina Ha, but I'm Miguel Reguero, and on behalf of CME Outfitters, I'd like to thank and welcome you for joining us for today's educational activity called Unlocking the Value of Biosimilar Therapies in IBD. Today's activity is brought to you by the AGA as well as CME Outfitters. As many of you know, CME Outfitters is an award-winning accredited provider for continuing education for clinicians worldwide. Today's CME CE activity is also eligible for ABIM mock points. Once you complete today's program, it's important that you provide your ABIM ID and birth date in the evaluation. So again, please submit your ABIM ID and birth date in the evaluation. CME Outfitters will then submit your mock points. If you would like to claim this activity as a CME for MIPS improvement activity, follow the steps that are described on the slide here. So for clarity around biosimilars in this presentation, we will be using brand names. And I think that you're probably familiar with this. So we will be using brand names, which we usually avoid in CME programs. But because of the discussion of biosimilars relies on us to use the brand names, you will hear us say that, and we'll remind along the way while we are doing this. So again, I'm Miguel Reguero. I'm chair of the Department of GI, Hepatology, and Nutrition at the Cleveland Clinic, as also the vice chair of the Digestive Disease and Surgery Institute and professor in the Clinic for Lerner College of Medicine. I'm also joined today, and everybody knows Tina, and you've seen her already throughout this conference, but Tina Ha is Associate Professor of Medicine, the F. Wijwa Foundation Inflammatory Bowel Immunology Research Institute at Cedars-Sinai in Los Angeles. Welcome, Tina. It's good to see you again.
Great to be here. Thank you.
So we list our disclosures here. Here are my disclosures. And then Tina's disclosures. And then let's go through the first learning objective. This is really the first learning objective is to understand the key terminology related to biosimilar products, such as interchangeability, substitution, and switching. And I should say that biosimilars are really here to stay. And you'll see the number of biosimilars that are in the pipeline. And I think probably most of you have had familiarity already with biosimilars. But there's a lot of confusion around these terms: interchangeable, substitution, switching, extrapolation, and we're going to go through this tonight during this conversation. I will also tell you that Tina and I are going to bounce back and forth in terms of presenting slides, but really also having a discussion. And we have some case-based approaches to biosimilars, which are common scenarios that we've seen in our clinics, and I'm sure many of you have seen in your clinics as well. So Tina, before we get started, maybe we can get to our audience involvement. So why even have a discussion about biosimilars? And I think this is an interesting study that I'm going to present and some interesting data. Look at the left-hand side of the slide here for a minute, and just to ground you into what the lines are. The UC Biologics is kind of the dark blue line at the very bottom. UC immunomodulators is kind of that green line. UC 5-ASAs, that's the very top line. And then UC corticosteroids is the dark black line. And I think if you look at the left-hand side, it's pretty obvious. If you go back to 2007 through 2015, there's been a leveling off or a slight decrease in 5-ASAs and an increase in biologic agents for the treatment of ulcerative colitis. If you look on the right-hand side, outpatient IBD medication cost, probably not surprising. You can see really the immunomodulators and the corticosteroids at the very bottom. 5-ASA cost has also gone up. About 50% of our patients with ulcerative colitis do just fine with 5-ASAs, but the costs have gone up. But you've seen a really exponential cost in biosimilars for the outpatient IBD medications. So biosimilars increase access to biologics, and why is that? And maybe this is the kind of obvious statement around cost, but let's consider the first biologic drug, monoclonal antibody, was approved in 1998, and I remember that because a couple years before I'd come out of fellowship and we had no biologics at all. So the emergence of monoclonal biologics really hit the scene, and these became a definite improvement in our treatment. And then over time, if you go to the right to 2013, was the first monoclonal antibody biosimilar for Infliximab. And again, I'm going to use a brand name specifically for Remicade that came out in 2013. This did have cost savings associated with it, although Tina and I can talk about cost savings in terms of where the cost savings is. But nonetheless, there was cost savings related to this, which did ultimately increase our patients' ability to have access to the biologics. And then moving further to the right, so from 2017 to 2026, biosimilars do have the potential to reduce overall spending by nearly $54 billion. So this is really, in essence, a money equation in terms of the biosimilars. So Tina, I'm going to turn it to you and have you teach us a little bit about biosimilars, the names, also comparing biosimilars to conventional drugs. So let me turn it to you and we'll bounce back and forth as we go through this.
Great. Thank you so much. So before we talk about biosimilars in particular, let's take a step back and really understand what differentiates biologics from conventional drugs. The first thing to know about biologics is that they're actually complex protein-based constructs that are produced by living cell cultures. And these have high molecular weight. And the way that they're manufactured, because they are such complex heterogeneous structures, is a lot more challenging. If you think back to your biology days, protein structures start first with amino acids, and then there's a series of secondary, tertiary, and quaternary folds that result in the actual compound. So creating biologics are strongly process dependent. And you have to keep in mind, even though we're talking about biosimilars and their relative similarity or high similarity to the originator products, from a batch to batch of the same actual biologic compound, there will be some slight variability because of these protein constructs. What also differentiates them from the standard agents are that these are unstable. That's why the infusion-based biologics and the injectables do need to be refrigerated. Now let's compare those to pills, tablets, or small molecule drugs, chemicals that we're used to taking by mouth. These are produced by chemical synthesis, and they're much smaller in molecular weight. They have a well-defined structure and manufacturing process, and they are mostly process dependent, but these are also, because they're completely characterized chemicals that are compounded into tablets or capsules, they are shelf stable. So let's move on and talk about, well, what are biosimilars relative to biologics? So a biosimilar is essentially a copy of a commercially available biological agent that is no longer protected by its patent. And that's how I would actually encourage you to describe it to a patient, that it is a copy of an agent that's lost its patent. However, what you also need to know is that process of getting it approved and on the shelves is actually much different than just for a generic. It's undergone rigorous analytical and clinical assessments in comparison to the reference or the originator product. And the pathway to approval does require a whole host of steps, which we'll talk about later on, in order to prove that it's truly very highly similar. And I like this quote at this statement at the end. Like fingerprints from identical twins, the biosimilars are very highly similar to their originator product, but they're not exact carbon copies. However, those differences that are present are not clinically meaningful. So they're very highly similar. That's why they're called biosimilars rather than other terminology, which we'll discuss in just a little bit.
And actually, Tina, I'm gonna interrupt for a second because I think this is really an important point about biosimilars. And people often, and you're going to get to this, I think, people often confuse this statement with generics, and this is actually not generic. And I like the identical twin and the fingerprint. I also like something you said a minute ago, that we don't think of it this way, but again, I'm going to use a brand name, Remicade, from 1998 to the Remicade today. We don't think of this, but it probably is a biosimilar of itself because it has gone through so many manufacturer changes over time. But I think you're really driving home that now clinical meaningful differences, and that's really the key. So here's the generic drugs. This is maybe a nice segue for you to talk about this now.
Yeah, so I think it's really important as we're talking about biosimilars to our colleagues, our healthcare team, as well as the patients, not to interchange the word biosimilars and generics. So in order for generics to achieve FDA approval, all they really essentially have to do is show that it's the same active ingredient, same dosing strategy, and the same route of administration. And so that's really what defines a generic drug. But also we have to keep in mind biosimilars are not the same thing as second generation or biobetter agents. So second generation biologics are structurally different from the original licensed pharmaceuticals. And they're intended to improve performance while preserving the mechanism of action. So to put this in layman's terms, basically, if we think about anti-TNFs, Infliximab was the first anti-TNF agent that was approved for Crohn's disease and ulcerative colitis. However, we have other anti-TNF agents, such as adalimumab, certolizumab pegol, and golimumab, which work the same mechanistically because they're anti-TNFs, but their protein structures are very different than the originator, Infliximab. So these are not biosimilars. Biosimilars, again, are like those identical fingerprints or a copy, but the differences between the biosimilar and the originator are just not clinically meaningful.
Yeah, and actually it's interesting you mentioned that word biobetter, because this has actually come up as well. If you do improve upon a biologic that we have today, that has to go through an entire new FDA approval. So biosimilars being that fingerprint and identical twins, where essentially we have to use the word similar, not better, and not the same. So very good. So tell us, how do these get developed? And I know this can be confusing, but I've heard you present this before, and you do such a nice job of educating everybody. Why don't you take us through the development process?
All right, so let's do the best we can. So according to the FDA, in order to show that an agent can be used as a biosimilar, you have to actually show what's called the totality of evidence. So you first start with the product itself. And the first step is the technical development. So you're essentially cloning the reference product, and then you're constructing, purifying, and validating that that protein construct matches as closely as possible the molecular profile of the originator. And that's just creating the biosimilar. So that's step one. Then you actually have to confirm the biosimilarity using several steps. There's a stepwise approach. And within those steps, first, you have to actually demonstrate pharmacokinetic and pharmacodynamic equivalence. What do those terms mean? Essentially, if you break it down to what the drug does to the body and what the body does to the drug has to be the same with the biosimilar as it is with the originator product. And you also have to confirm efficacy and safety in tailored clinical trial studies, phase 3 studies. And once you can show that the efficacy, the safety, the immunogenicity, the pharmacokinetics and dynamics are the same in a disease state that the originator product is already approved, then extrapolation may be possible to some of the other disease states where the agent is already used. So to kind of step it back to looking at Infliximab, if you can show that Infliximab is effective in, let's say, rheumatoid arthritis and ankylosing spondylitis with a biosimilar, then you may be able to extrapolate that data to Crohn's disease, ulcerative colitis, and psoriasis.
So one thing actually, we're going to get to extrapolation for a minute, but the idea of immunogenicity, I think we have a case, and actually a few people asked earlier in the Congress when biosimilars came up, what's the deal with immunogenicity? Maybe say that one point again, because I think that's so imperative.
Yes, and so the studies the clinical studies for the biosimilar have to show that there is no increased risk or no differences in immunogenicity with the reference product or the biosimilar. So at the end of the day, the biosimilar Infliximab immunogenicity rates are essentially the same as the immunogenicity rates for branded Remicade.
Common question, Tina, for you. If you have a biosimilar, say for Remicade, for Infliximab, can you use the same assay, the same therapeutic drug monitoring assay that you would use for Remicade for the biosimilar?
Yes, because remembering that structurally they're nearly identical. So you can absolutely use the same strategies for therapeutic drug monitoring, the same assays as you would for the reference product. So your same clinical principles that you would use for the branded agent should apply for the biosimilars.
Great. And this is probably one of the kind of most important themes on extrapolation. And talk us through extrapolation, because this is one of those terms for definition and kind of what does that mean for the audience or for those out there using biosimilars?
So we're going to break it down to the strict definitions in this slide, and then maybe we'll go into the example with Infliximab on the next slide. So with extrapolation, you have to have a clinical trial in one indication where the reference product is already used. So for example, rheumatoid arthritis. And you have to show similar safety, efficacy, immunogenicity, PK/PD in that particular disease state. And if you can show that there's essentially no difference, no clinically meaningful differences between the biosimilar and the originator product for that already approved disease state, then you can extrapolate to other conditions where you already use the reference product. And the reason why extrapolation is possible, and it's something that's considered, is because you cannot have a biosimilar undergo a randomized control trial for every single disease state, particularly when the likelihood that you're going to see a different result is pretty low. Now, obviously, we have to account for there are different nuances on how we use biologics, how we dose, how we optimize from disease state to disease state. But at the end of the day, if you can show that it works for one disease state, you can likely extrapolate to others.
So I guess just to say this again, because this is important and you're going to get to this, I think, in a minute. So if a biosimilar, say for Remicade, Infliximab, or for adalimumab Humira is approved for rheumatoid arthritis, you're saying that essentially by FDA guidance, the extrapolation of the data, because it's a biosimilar for rheumatoid arthritis, can then be extrapolated for another labeled indication, which would be something like Crohn's or ulcerative colitis. So it's essentially saying, because we know it's working in a rheumatoid arthritis, it's a biosimilar, we can extrapolate that to Crohn's and ulcerative colitis. Is that correct?
Exactly.
Perfect. So keep going with extrapolation, and now we get into a lot of these fancy letters.
Yeah, and I think that the most, as confusing as this terminology may be, the letters are probably going to be worse. So really, our first, at least, foray into extrapolation for the inflammatory bowel disease patients was with Infliximab biosimilars. So there were two important studies looking at the Infliximab biosimilars, Planetra and Planetus, which looked at rheumatoid arthritis in Planetra, which was a phase 3 study, and a phase 1 study looking at Infliximab biosimilar versus originator product for ankylosing spondylitis. So between those two studies, looking at pharmacokinetic and pharmacodynamic data, looking at clinical efficacy and safety, as well as immunogenicity, they found no differences between the originator and the biosimilar. So because these are already labeled indications for Crohn's disease, ulcerative colitis, psoriatic arthritis, plaque psoriasis, and pediatric Crohn's disease, they were able to extrapolate that the results would likely be the same for these states, and that's why we have biosimilars for these labeled conditions approved.
Right, except like you point out on the slide here, because it wasn't approved for pediatric ulcerative colitis, you can't get the extrapolation for a disease that was not approved by the innovator product.
Mm-hmm. So the last part of talking about the biosimilars approval process, because we talked about creating the actual protein construct, we talked about the clinical studies to demonstrate similarity in terms of the meaningful endpoints, and then what's important is the post-marketing pharmacovigilance. So what they mean by that is the companies that create the biosimilars, they do need to follow their patients over time and the biosimilars over time to look for adverse events and drug-related problems. This isn't unique to biosimilars, by the way. This is actually true for any biologic that's out there. They need to ensure and show that they're using consistent manufacturing methods, they're monitoring for immunogenicity, and most importantly, they need to make sure that their naming of the biosimilar is distinct so that patients, providers, infusion centers, and specialty pharmacies are able to distinguish the originator product from the biosimilar. So the way that the FDA decided to make that easier was to make it more confusing. So what they did was they actually have a 4-letter suffix that's attached to the the biologic drug, such as Infliximab-DYYB. Now you can ask, what do these letters mean? Is this some sort of specific code like expiration dates? And the honest truth is they're actually meaningless. They actually have no point, but they're random, unique four-letter sets that help differentiate the various types of biosimilars. Now it's important to know for already approved biologics, there is no four-letter suffix. However, moving forward, as new biologic compounds are being created, whether or not they're originators or biosimilars, they will actually all have those four letters. So we may in the future need to be able to have a table where we can identify which one's the reference or originator and which one's the biosimilar.
And that's going to be incredibly confusing. I think this is actually something that my patients quite often ask about when they see the DYYB, or for adalimumab, the ATTO. And I think you've said this, but just to drive this point home, these are not letters that mean anything. In fact, they have to mean nothing. So even though the suffix is there, it's not like you can try to figure out some catchy way to remember them. These are truly meaningless, and that's how these suffixes are designed. And as you said, I think in the future we're going to see that as well. So let's go on to the biosimilar versus interchangeable. So, we had this as the poll question, and get into what interchangeability means, and then also, are there interchangeability in the United States? Can we do this right now?
So I think the most important part of this slide is what's at the very end with that warning sign. So what interchangeability means is that an interchangeable product can be substituted without the intervention of the healthcare provider. So essentially at the pharmacy or the infusion center level. And that's what interchangeability means. It doesn't necessarily mean that you were in the hospital, you received branded Infliximab, and then your next dose after discussion with the provider was switched over to a biosimilar. That's a different discussion. Interchangeability means without the notification of the patient or the provider, this can be switched at the pharmacy level. So the FDA states that in order to determine that a biosimilar is interchangeable, they do recommend and they expect that there's additional clinical data. So when we talked about the totality of evidence that needs to be presented in order for an agent to be FDA approved as a biosimilar, that's only essentially as a new start for an agent. But if you're talking about switching back and forth from one agent to a biosimilar back to the reference, you actually need additional clinical data, which is an important point.
Great. So interchangeability is confusing. Let's go back to the poll question because we can see what our audience says. And then Tina, maybe I'll ask you about what you think as far as the results to the poll question. And then we can also get a little bit, there are a couple questions about this idea of interchangeability as it gets into some of the issues of head-to-head studies and have there been any between, say, Humira and biosimilar adalimumab Humira, and then Remicade and biosimilar. So let's go to the poll question first. And thanks, Tina. That information was absolutely phenomenal. So Tina, when I looked at the results, how many biosimilars approved in the United States are interchangeable in IBD? Here were the results from the poll. So prior to your lecture, 41% said all biosimilars are interchangeable by definition. 23% said, I don't know, so still you need to tell them the answer. 15% none are interchangeable. 11% said around 50% are interchangeable. And then 10% said around 25% are interchangeable. So Tina, what do you think?
Well, it's about the nuance of the question, if you will. So technically, by strict FDA definition, the answer is none. But we all know in our clinical practices, we do have non-medical switches. We do have interchangeability occurring at the healthcare provider level. Sometimes it's dictated by the insurance. But so the reality is, by strict FDA criteria, no. But does it happen in clinical practice? Yes.
Yeah, so I think certainly this will never show up on a board exam or any exam that's meaningful because it's incredibly confusing. But again, as Tina said, by the strict definition, the answer is no. Tina, there are some questions that are coming in, and we'll leave plenty of time at the end. But one came in that specifically, actually, a couple of people are asking a similar one. Are there head-to-head studies between, say, biosimilar Remicade with Remicade Innovator, the original Remicade? Do we have anything to guide us there?
Yes. So there are actually a number of studies that have been published within the IBD space for both Crohn's disease and ulcerative colitis that have looked at patients who, for example, are new starts to Infliximab, and they received either the originator Remicade or they used the biosimilar. And the overwhelming amount of data is that there really is no difference in terms of those key components: efficacy, safety, immunogenicity, and to a certain degree, durability or persistence of treatment. So there are a number of studies within the IBD space looking at new starts. There are also a number of studies within the IBD space that look at switches, non-medical switches, and we'll cover some of that data. But that's also the case. There is increasing data also for adalimumab in the IBD space as well, comparing new starts as well as some switches.
Great. And actually, we're going to get to a case in a minute, but I was a little bit delayed on the post-polling question. So actually, Tina, you did a great job of teaching people, because the biosimilar interchangeability, the none answer went from 15% up to 36%. So I think you did a great job teaching that technically by the FDA, there's not interchangeability. But obviously, this still remains a confusing topic, and we get it as well. So let's talk about a clinical scenario, and I think these will really drive home some of the points. We have some other clinical scenarios sprinkled throughout as well. So this is a new patient, Maria, who recently was diagnosed with moderate to severe Crohn's, and you're starting her on Remicade. And again, specifically the reference Infliximab. So Remicade, as we've known it since 1998. However, here's to your point, Tina, that you just made. Maria's insurance company approves an Infliximab biosimilar instead. So are you comfortable with this decision, and how do you explain it to Maria? So I guess, Tina, this comes up all the time in our practice. What happens when the patient calls you and says, hey, they're switching from Remicade to one of the biosimilars? Are you okay with that? And then what are you telling your patient?
Well, in this particular instance, it's a new start. And so what I would encourage, the audience, the listeners in the audience to counsel themselves as well as their healthcare team, the patients, is to actually change your nomenclature. Instead of using branded names for all the biologics, we have to actually be familiar using the reference names, such as Infliximab, adalimumab, certolizumab pegol, vedolizumab, ustekinumab, etc. Because if you get so fixated on using a trade name, then if you have to use a biosimilar instead, then there's automatically this perspective from the patient, wait a second, you're changing my therapy. Whereas the overwhelming data show that you're giving them Infliximab, whether it be Infliximab that's called Remicade, whether it's Infliximab that's called whatever the trade names are for the biosimilars. So for a new start, I think we actually have to change our language, knowing that biosimilars are here and they're here to stay. With respect to switching, I think it's all about making sure that we actually introduce the concept. Now, it's tricky because sometimes these changes are mandated at the insurance level. So now what we're trying to do is we're trying to recognize patterns, which insurers may actually use a biosimilar preferentially first, which insurers may not, and prepare the patients before we start or as we renew to say, listen, you may be seeing additional 4 letters next to your Infliximab infusion bag, and it's still the same agent, we know. But part of it is all about the communication and how we define things.
Great. Yeah, no, I actually like the way you explain that, because I think in a way when a patient comes in, I said, you've been on Infliximab for however many years, and you're essentially switching to Infliximab. So it's, again, this biosimilar idea. I think the letters are confusing. The other thing that you said earlier that I think really helps is that I tell my patients that the drug monitoring for these is the same, because you're essentially measuring Infliximab in the blood and antibodies to Infliximab, or adalimumab in the blood and antibodies to adalimumab. So I do the same thing as you do. Obviously, there's always confusion and concern when something new comes out, and unfortunately, I think that this is a confusing topic. So let's switch now to our second learning objective, and in a minute we're going to get into our second polling question. And Tina did such a great job with the first set that in a minute I'm going to turn the slides back over to you to present. But let's go through the second learning objective, which is really to assess the efficacy and the clinical trial data and consider real-world efficacy of safety in biosimilars. So here's another audience response. So please, everybody take a minute to answer this. And then I guess as we wait, in essence, Tina, this is kind of a loaded question, and people may not realize this, but if you have diabetics who are, say, on insulin, you know, there's been this biosimilar to insulin for a while, and we don't think about this in GI or in IBD, but probably without knowing, I don't know, what do you think? There's a pretty high rate of biosimilar use in general, right?
Yes, and and biosimilars have been out in the hematology and oncology field for quite some time too. I think anytime you make a change, there's this period of adaptation. But I think that as we all become more familiar with biosimilars, we'll view it kind of the same way that if you think about it, I don't use the same brand of acetaminophen every single time that I, go to the store. So I think that as we become more familiar and comfortable with the concepts of biosimilars, the scariness will go away.
Yeah, and so the polling results did come in, and it's interesting. I think probably we could have predicted this, but the majority have only 1 to 24%, and then next was about 25 to 49%, and then third was none. So only a few people actually had the majority of their patients. Now, I will say that our institution in Cleveland 2 years ago switched to a biosimilar Remicade. And so I think probably many of us, if we're practicing in the system that I'm in, would say 100% because by this point we've actually seen it. So let me go through to just show you overall the IBD biosimilars in the United States and confuse everybody even more, but to drive home the point that Tina made, where we're really going to have to get used to using the term adalimumab, infliximab, and those are the two that I'm pointing out now because the others have not yet, but I'm saying yet because I think they will over time. And you can see, for example, adalimumab has 6 listed on the left-hand side, and then I'll give the example adalimumab ATTO, ATTO, which is Amjevita. So again, this is going to be a biosimilar to Humira, to actually Humira innovator product. And on the right-hand side, Infliximab biosimilars, you can see 4. I think some of your systems or people out there may be familiar with Infliximab DYYB, which is Inflectra, or Infliximab ABDA. Again, the letters mean nothing and they are impossible to remember, which is Renflexis. And Renflexis and Inflectra are both commercially available. Apsola is also commercially available. So those are the ones that you probably have heard of. So the ones that have asterisks next to them are commercially available. But stay tuned, and at the bottom of the slide, I think appropriately, many more to come and many more to have commercial availability soon. So let's go to this case, Tina, and let's see what you think about Jessica. So Jessica has been on Remicade reference infliximab for 14 months. Her recent labs reveal that she has an increase in anti-infliximab antibodies. And she recently heard about a new therapy, and she had done well with Remicade initially, but now she's developing antibodies. And she wants to know if Apsola, infliximab AXXQ, is something that you can switch her to. So what do you think, Tina? She's been happy with her Remicade. She's now getting antibodies. And she said, you know what, can you switch me to a biosimilar Remicade? What are your thoughts there?
Well, the first thing I would say is, had I not known that Apsola was an infliximab biosimilar, I may be thinking, wow, there's a new biologic out in the market that I don't even know about. So I think it's going to be very important as more of these are approved in the market and they have trade names in addition to their reference names. It's going to be very hard to keep track because initially it sounds like she wants to be on a fancy new medication, but it's actually the biosimilar to infliximab. So you would use the same rationale as to, with respect to the biosimilar as you would with her current dosing of infliximab. You'd ask, okay, what are the levels? And is this something I think I can overcome by using combination therapy with an immunomodulator, dose optimizing, or if the antibodies to infliximab are high enough and the levels are undetectable, you have to switch to a different anti-TNF or potentially a different mechanism of action. So when you have antibodies to reference infliximab, It's essentially that you have antibodies to infliximab. So the likelihood that you'll have an infusion reaction or an adverse response to the biosimilar is going to be quite high.
Right. So the bottom line with her is we tell her that although it sounds appealing, we do not do that, and we would not switch her to biosimilar infliximab for the very reason you have. And again, I think your point about if we call it all infliximab at some point, it's essentially, I think it's going to be easier for people to comprehend. So thanks for driving that point home. Tina, I'm going to now have you take us through the clinical trials. We're also going to get into some of the switch studies, a lot of the questions that are coming in around switching. So I think the next 3 slides are probably some of the more important for this topic. So why don't you take us through the clinical trials in IBD?
Yeah, so I think it's really the interchangeability and the switching that's going to be most applicable to our clinical practices. So when we were talking about the process for approval and extrapolation, it was really based on clinical trials looking at disease states that are usually rheumatoid arthritis, ankylosing spondylitis, psoriasis, or psoriatic arthritis. And then you can extrapolate based on that data. However, you can't assert interchangeability based on that data. So we need information that's based within the IBD space or within those disease states where they actually have set switches. And so let's talk a little bit about how some of these studies are designed so that we can demonstrate interchangeability. So there's actually several different types of studies. The first is one that's actually very commonly used, where you have 2 groups of patients. One is on the reference or the originator biosimilar, and the second group is given the biosimilar. And at a certain point, whether or not it's 8 weeks, 12 weeks, 24 weeks, 52, the patients who are on the originator transition and they switch over to the biosimilar, and you look for outcomes. Another is where you start with, and that's a transition study or a single switch. There's also studies where they do multiple switches with a reference drug and a biosimilar based at set time points that are currently under investigation. So I think rather than just looking at how these studies are designed, why don't we go through a couple salient clinical trials?
Yeah, and actually before you go through the clinical trials, because I think that the NOR switch and some of the studies we're going to show, it's the single switch. If everybody out there, the single switch is probably what we're going to present and are most familiar about. But I bet most of you are going to ask about multiple switches, because multiple switches comes up, and if you have a batch of, say, infliximab, as we said, there are 3 commercially available, could there come a time when there's almost pooled switching, where you're on one time, then the next you get a different, then the next you get a different, although they're all biosimilars. But right now, that kind of the middle row that Tina mentioned, the single switch, is really important. So Tina, talk us through the transition, the single switch, NOR switch. What are the data? What did that show?
This is actually one of the more important studies that looked at a single switch from reference infliximab to a biosimilar CTP13. So before we kind of talk about the data and the results, you kind of have to understand how this study was designed. It's a phase 4 multi-indication. So it was essentially looking at patients who are on infliximab for any of the approved conditions. And this was performed in Norway. In order to be included in the study, patients had to be on a stable maintenance treatment for at least 6 months. So our patients with ulcerative colitis and Crohn's disease weren't necessarily in a stable maintenance dose in remission. They were just in a stable maintenance dose. And so when you look at their data and their primary outcome was disease worsening. And the outcomes that they used for ulcerative colitis and Crohn's disease were clinical disease indices, changes in the partial Mayo score or the Harvey-Bradshaw index. So as we're looking at this data, we have to remember that clinical indices don't necessarily mirror the objective findings that we look for in let's say, a treat to target type of practice. So at face value, you could say, that curve of the Crohn's disease is not looking so favorable. But if you really dissect the study population, only about 60% of the patients were in remission by Harvey-Bradshaw index at the time that they were enrolled. So some of this could also reflect just the natural history of disease progression and response to therapy, even if they maintained on their reference product. But that's how this study was designed. But overall, in their aggregate data, looking at all these disease states, they really showed no differences that favored either the originator or the biosimilar.
Yeah, and I think the important part of this too is that this was really, I think, in IBD at least, the first study. It looked through 52 weeks. But now take us through the kind of the longer term and also the other diseases as well. So talk about this, and then I'm going to ask you a couple of questions that are coming in from the audience.
Sure. Now, it's also important to note that in Northwich, even though the primary endpoint was disease worsening at 52 weeks, they also didn't see any differences in immunogenicity in terms of trough levels, anti-drug antibodies, or adverse events. So in terms of the Northwich open label extension, basically it was a 26-week extension study after participants have completed week 52, 52 weeks of the study, and all patients were essentially treated with the biosimilar. It's the same outcomes that was in the original Northwich study. The primary outcome was disease worsening at week 78. And they basically saw the same types of findings, that there were no differences in disease worsening, drug levels, antibodies, or adverse events. And again, you can look at the curves and it can say that it favors the switch towards the biosimilar CTP13. But again, you have to think about sample size, the study was powered, and what their patient population's disease activity was at baseline.
Right. And so, Tina, let me ask you kind of a loaded question, but you and I have both faced this in our own practices. So you're in an institution, the patient's on, say, Remicade, and based on your institution and the insurance, they switch to biosimilar Infliximab. Are you comfortable with that? Are you okay with that?
Mm-hmm. Yes, so I think that we actually have a pretty astounding amount of data looking, especially for biosimilar Infliximab, growing for biosimilar adalimumab, that I would. And but my same principles for how I would optimize and monitor with branded Infliximab apply for biosimilar. So let's say that because for a lot of these studies, the switch occurred when patients were on a stable dose, in a stable remission or stable response. But let's say somebody's disease was acting up a little bit or their levels were a little bit low. I would optimize their Infliximab, use therapeutic drug monitoring, and use the same treat to target endpoints as I would with the originator, with the biosimilar. It doesn't change my practice because I'm prescribing the mechanism of action anti-TNF via infusion.
Yeah, so I think you and I practice very similarly. And actually, Rachel Winter and Jessica Allegretti had some great questions that I think they're along the same lines, but maybe I'll ask Jessica's because I think Rachel's gets to the same point is, when do you feel strongly about fighting or arguing with the insurance company when they're mandating non-medical switches? Is there ever a time when you would do that? And I think Rachel had some similar kind of questions to that theme.
Yeah, in the past, what I would, if you would have asked me maybe a year or two ago, I would have said, maybe I would take pause if somebody's pregnant before making any changes from branded to biosimilar. Or if we haven't established what their set dose is going to be, let's say they're not on a stable maintenance dose. But my concern is this, in the ideal world, I wouldn't want to change until I knew they were at a stable dosing or in a stable remission. However, oftentimes, fighting the insurance can result in delays in the treatment. And we know, especially for Infliximab, the immunogenicity can be quite high, particularly when they have active disease and their risk of forming antibodies is greater. So I have to weigh what is the likelihood that I can get this overturned versus what is the likely harm of giving them the biosimilar optimized the same way I would the reference product. So my perspective has actually changed where I'm a lot more comfortable switching somebody to a biosimilar as long as it keeps them on the same dosing schedule and gets the job done.
I agree. And I'll just simply add to that, now that I've had 2 years of doing this, and at first I think everybody had a bit of concern in the first 3 to 6 months, and we're going to actually report out our data because between derm room and GI, we have a number of patients now who've switched because of this. And so far, just the bottom line is I haven't seen a problem, and I'm comfortable with that. So Tina, let's go to this clinical scenario. So I think this is kind of what we just talked about. Your patient, Andrew, was switched to an Infliximab biosimilar. How do you help them ensure that the drug's working for them? And then I guess more importantly, do you do anything differently? Do you pre-medicate? When they switch, do you automatically check levels if they haven't had levels in a while? Is there something that you do differently when they switch?
So in terms of clinical practice, no, because I would use, if I wouldn't pre-medicate with a reference product, I wouldn't pre-medicate with the originator unless there was an issue such as an infusion reaction or low levels of antibodies, and I thought it was necessary. Same thing with therapeutic drug monitoring. I would use it the same way. The only thing in my clinical practice I would change in this scenario is, and I think we're going to talk about it later on in the last section, is doing my best to avoid the nocebo effect. Because if you make it like, oh my God, your insurance sucks, we have to change you from reference to the biosimilar, what a disaster. I mean, but take it. That's automatically going to plant the seed of failure. But if you reassure, you're getting Infliximab, you need to stay on it. You're doing great. We're going to monitor you the same way. You have to make sure that the buy-in is from all parties, not just as a provider, but also with your clinical care team that's responsible for getting the infusion set up, scheduled, and approved.
It's so funny that you say that. We'll get to nocebo in a minute, but when we first had this in Cleveland, we had, I think a lot of our team and a lot of our physicians were concerned, and I think whether it was subconscious or conscious, there was a little bit of that statement that you just made, and we had people have what we thought were reactions. We actually prompted us to check more therapeutic drug levels, and we didn't see any problems. So now that we're all comfortable with it, and it's not to be dismissive, so it's not like we can say, oh, there's no big deal, don't worry about it. But now that there's a much more familiarity, I think we're okay. So Tina, why don't you go ahead and tell us about the switch data with adalimumab? And one of the questions that came in that we'll go all the way to the end now with the slides, and then we'll leave plenty of time. But somebody asked in the audience when adalimumab is going to be approved for biosimilar or commercially available. I should say commercially available. But why don't you take us through these data, and we'll keep going.
Yeah, so, because this is a switch study, so before talking about the switch study, since we haven't really covered too much about adalimumab, I just wanted to highlight a study that we don't necessarily have a slide on, but was presented at DDW as well as at some conferences last year. And it's an important one for us to know as we're counseling our patients when adalimumab biosimilars are commercially available. And that's the Voltaire study. And basically what they did was they took a group of people and they randomized them into two groups. They were new starts for adalimumab. They received either the reference product or the biosimilar. And the primary outcome was just looking at response at week 4. And they really saw no difference in response rates at week 4 for patients who were given the originator adalimumab versus the biosimilar. Then they had the patients continue on with the therapies that they were randomized to, and they didn't see any safety signal differences at week 24. Now, what we're going to be looking forward to is that at week 24, patients who were given the branded adalimumab are going to have a switch to the biosimilar, and then we're going to look at outcomes. But this is a data from a switch study, but it's actually a combination of a switch as well as a new start. So they looked at patients. This is an observational study, small numbers, but the graph on the left basically looks at new starts to a biosimilar to adalimumab. And this graph shows persistence or durability of treatment. So most of the patients who started the biosimilar to adalimumab, who are naive to treatment, were continued on therapy at 7 months, and they didn't see any real differences in clinical response or remission rates. They also had a subset of patients who were on a stable dose of originator adalimumab, and they were switched over to the biosimilar, and they also saw similar rates of persistence of treatment. So now you can argue this may not necessarily be the best endpoint to show comparability between the biosimilar as well as the originator in terms of efficacy and safety, but persistence of treatment is actually an important endpoint because ideally you're not continuing a treatment that isn't working.
Yeah, so I think the important part of this, as you mentioned, we talked a lot about Infliximab, but we see the same thing with adalimumab biosimilars, both in new starts and in switches. And here's some bottom line as we then transition to the third and final learning objective. So I think these are, and a lot of you are chatting in some great questions. I think some of this may answer that. Data support similar efficacy in new starts. So absolutely no problem in new starts. Also, there's growing evidence now to support safety and efficacy of switching for patients. And again, I think that's the single switch data that Tina presented, and certainly I'm very comfortable. I think the third bullet point is what probably, Tina, when we get to the Q&A, we can have the discussion, is this data are limited and reverse or multiple switching or cross-switching amongst biosimilars. And I think that's what everybody, and a lot of the questions we're getting are going to be around that. And I think everybody's realizing that's probably where we're headed. There were some presentations this year at DDW and some of the European meetings on multiple switching. My bottom line, we're not going to show data necessarily, is that there was no problem, but I agree that we need more data on the multiple or the batch switching. So let's go to another clinical scenario, which we've already talked about, so we can decide if we want to just briefly touch on this and move on. But again, that your institution, the insurance, switches everybody from Remicade to Infliximab biosimilar. You're getting lots of calls, which, like I said, we did for that first 6 months. I think, Tina, you already said this, but again, just to remind the audience, that next dose, going from Remicade to then biosimilar Infliximab, that next dose, are you doing absolutely anything different for the infusion center, pre-medicating, or are you just saying, you know what, whatever you were doing for your Remicade, you get that for the biosimilar Infliximab?
Now, the only thing that we're doing differently is making sure that they signed up for the right savings program, if it applies, but otherwise it's the same.
All about the money.
Yep.
All right, so let's go to our final learning objective. I mean, I think the discussions we had so far have been outstanding. And this final learning objective is how we improve patient-provider communication around the use of biosimilars. We do have another audience response question. So what would you answer? What's your answer to this?
You know, the honest answer, it should probably be a combination of A and B. Yeah. But the reality is, you know, as we submit to insurance, we're submitting for Infliximab, we're submitting for adalimumab, and they have to spit back to us what they're going to accept or reject. Thankfully, it's usually a rejection, so it gives us time to kind of, prepare the patients. But I do think that systems-wide, we need to do a better job, both from the healthcare systems, the insurers, communicating whether or not a patient, what's the first line. It's very simple to say that it's an anti-TNF or it's Infliximab, but once we know what is the first line for that particular insurer, it makes it much easier to be transparent about the process and the counseling.
Great. And the poll came in, it's interesting. I think if we had insurance companies up here, maybe we would have had a different response. But the majority answered that physicians, followed by NP, PA, and nurses. So I think that's great. I mean, that's great to hear. I think that's what it should be. I think in the future, we may even have some of our specialty pharmacy and our pharmacists that we just had a pharmacist to our IBD group. We'll probably also be doing a lot of the education around this. So let's get to patient voice and gaps in patient-provider communication. I just want to, in a minute, I'm going to play the audio for this, but I think a lot of you out there have probably seen this. So speaking as a patient or on behalf of your patient community, please tell us how effective healthcare providers are in informing patients about the differences between biosimilars and generic agents in the management of IBD. So these are actual real patients that were surveyed, and I'll play what they said.
I have not had any of my rheumatologists discuss biosimilars with me, so they've never brought up that they exist at all, let alone the pros and cons of, using a name brand biological versus a biosimilar, which, considering the outrageous cost of most of the name brand biologicals, that's super frustrating.
I know the way that it was explained to me was that it was similar to a generic, and I at first took that as face value, and that was really all the information I was given.
Yeah, so Tina, you kind of hear there, and we're picking on rheumatology, but to be fair, this probably could have been gastroenterology. So this is another patient. Speaking as a patient or on behalf of your patient's community, how effective are healthcare providers in educating patients about biosimilars, particularly regarding the concept of interchangeability of biosimilars with reference biologics and how it applies to the safety, efficacy, quality, and biological activity of biosimilar agents. So
my own healthcare providers have not done any educating on biosimilars at all, let alone the interchangeability. More patients are learning about biosimilars from their insurance company than they are from their doctors.
Yeah, so Tina, what do you think? You kind of alluded to this already, and you heard a few patients' voices. Is this kind of what you're hearing in your practice, or what do you think about these comments?
You know, it's funny. I laugh because I see a lot of similarities between the biosimilar rollout as the COVID-19 vaccine rollout. You know, we find out that it's available to our patients after the fact. And so, you know, I think that there's certainly a lot of room for improvement in terms of how the various systems that are involved, the healthcare team, the insurers, the infusion centers, the specialty pharmacies, deal with the concept of biosimilars. But I can definitely appreciate, particularly for, we don't like change. It's very difficult. And so if somebody's doing incredibly well, and especially if they had a tumultuous course to get into a deep remission on their agent, I can understand hesitation about wanting to being switched over to a biosimilar. So I think it's really important that as a healthcare team and all the members that are involved in the authorizations and the approvals and the counseling process are familiar with the data and be able to say, listen, we're in the same way that we convinced people that it was okay to take Remicade, the same way that we can say, listen, an Infliximab biosimilar is okay. We have the data. It's been used. This isn't something that's a 2021 new construct. It's been around. And the more make patients feel confident with our experiences and how that's going to translate to them, I think that's going to matter the most.
And when you talk about patient concerns and some of the confusion, why don't you present this? I thought this was interesting. And then this gets into the nocebo effect. And then
yeah, so this is a very important study, and it really highlights that biosimilars, while it may be a concept that, you know, we may have heard of in talks such as these, the majority of patients actually have never heard of biosimilars. And, you know, this is, again, where the naming process, they have trade names, becomes a little bit more intimidating. And a lot of the patients' concerns center around the safety of the agent because it, at face value, looks like it may be a brand new treatment. They're also concerned particularly how their clinical course was pre-treatment before they were on the originator drug, that maybe they're getting a lesser product and it's not as effective. And they're also, and especially when you're dealing with names such as Inflectra, Remsima, especially Remsima, compared to Remicade, that it's a different agent that they're getting. And so that results in a nocebo effect of up to 10 to 15%. And I think that this is really, and the thing about these patient concerns is that they're all modifiable with the right background and the right education and open communication.
Yeah, and I think communication is key. And I really like this slide that you kind of help put together. So maybe talk about the nocebo effect. Like I said, I think we just have then a couple more slides, and then we're going to get to some questions.
I mean, we're all very familiar with the placebo effect, right? You think you get a positive outcome from a placebo treatment because you think you're getting something fancy and new. So the nocebo effect is kind of the similar, but a little bit more negative. So your perspective or your response to treatment is influenced by your preconceived notions and expectations prior to receiving the actual agent, even though it's essentially the same thing. So the good thing about the nocebo effect is that it's modifiable without using things like steroids. And so the way that we can modify the nocebo effect is really through these four essential components of this rectangle. And, but this translates not just to biosimilars, but it's with everything: treat to target, starting biologics, counseling about treatments in general. The basis is solid physician-patient communication, not just with the patient, but also multidisciplinary management, making sure all members of the health team are familiar with the concept of biosimilars, and really and trying to do the best to prophylax, prevent, and recognize the nocebo effect, and just learning and understanding what patients' concerns can be about making the switch, and doing your best to listen to keep the lines of communications open, have close follow-up, and assure them that everything else remains the same. All it is one change from one highly similar drug to another.
Right. And so talk to us about the stakeholders and what should we know about this?
So, you know, and I think you had mentioned it earlier, biosimilars are here to stay. So they're going to be part of our nomenclature. They're part of our therapeutic armamentarium. They fit the exact same place as the reference product. And why are these important? Well, there's four key things: additional options at a lower cost, savings and efficiencies to the health system, which may rub people the wrong way. They'll say, well, I don't care about saving the man the money. I want to make sure that it's not impacting my wallet. But increasing access to biologics, I think, is really key, and to help foster innovation by leading to new mechanisms of action. Because we know that the earlier we can start patients on steroid-sparing strategies, the better. We know that there has been limitations in access to biologics due to cost, due to widespread availability. So hopefully, as we have more biosimilars to Infliximab, adalimumab, and others, we can reach more people to ultimately result in better outcomes.
Yeah, I think, and just to tag on to that again, given my probably now 2-year experience with biosimilar, I think, and I don't want to say this uniformly because this may not be true of everybody, but I think it's actually been a bit easier in terms of access and getting approval and getting started. And to your point earlier, if we're really stuck and we're trying to go back and forth with the insurance company or the health plan and we're delaying, that doesn't help anybody. And I will say I'm very comfortable with biosimilars, but it's also something that, to your point about access, and I think when the pool increases, we're going to continue to see that.
And hopefully it results in changes in things such as step therapy, which we know is terrible. So by decreasing cost, increasing access, maybe we can get them the biologics they need faster.
And I think Crohn's Colitis Foundation has done a really good job as far as looking at step therapy and helping our patients. And I completely agree. Well, we couldn't go through an entire talk without COVID. So we'll end with this case and then we'll wrap it up, but then have plenty of time for a lot of your questions. So Tina, your patient Sandy was switched from Infliximab biosimilar, to an Infliximab biosimilar a couple of months ago. She wants to now get the COVID-19 vaccine, but concerned about the safety since the medication she's taking now is relatively new. Says she had been on Remicade for a long time. Now she's on Infliximab biosimilar. What do you tell her specifically? And then maybe broadly, what are you doing with your COVID-19 vaccines in your biologic patients on biologics?
Well, at face value, I would counsel her the exact same way I would counsel somebody who was on the originator Infliximab, that the indications, the safety, the presumed efficacy and safety data amongst our IBD patients is exactly the same. However, the reality is the nocebo effect is there. And for somebody who's been doing well on a reference product to make a switch, the only thing I would say, and it has nothing really truly to do with the safety of doing Infliximab and a COVID-19 vaccine close together, is I would probably space it out a little bit. So to avoid the blame of the first dose or the second dose with respect to the brand new biosimilar, only so that it doesn't color her perception of the safety and efficacy of the vaccine and the safety and the efficacy of the biosimilar.
Yeah, and I do the same. And I think the timing of vaccine related to the biologic therapy, first of all, like you said, whether it's biosimilar or not, that to me, it's the same discussion. I think that the point I'm making with my patients, so depending on where you are in the country, it may be difficult to even get the vaccine for many of our patients right now. But when the vaccine becomes available, I am recommending the vaccine for all of my patients. And I'm not looking at timing as much, but to your point and what you and I even discussed earlier, if they're going to get the vaccine the same day they get an infusion or same day they get their injection, it's maybe not a concern about efficacy of vaccine, although we can have that discussion separately as far as does immunity get impacted by the biologic. But it's more if they get a reaction or if they have symptoms to the vaccine, which are not uncommon, muscle aches, and like we would with a flu vaccine. You never know if it's the infusion biologic or the injection. So I think just practically speaking, I'm trying to separate, if I can, from the day of the injection or infusion. But again, I don't want to delay either. So let's go through some of the SMART goals. Then we'll wrap it up, but then we'll get to a lot of the questions, and we have till the half hour. So I'm really happy, Tina, that we're going to have more time for the questions here. So first of all, thanks, Tina. That was a phenomenal discussion. As always, you're so up to date on the biosimilars, and I really always enjoy, and I've learned from you as well. I'd really like to summarize 3 SMART goals now for everybody. One is to stay abreast of the recent clinical trial data on biosimilars, which Tina started, but there's more and more coming out every day. Also, recognize which patients in your own practice that you have a financial benefit from switching. Now, the financial part's tricky because we realize the savings doesn't go to you, it doesn't go to the patient, sometimes goes to the health system. But like Tina said, I think sometimes that actually will, as more biosimilars come out, make it easier. And then obviously we need to involve our patients in the decision around switching. So Tina, here's a question that came in, and a lot of people responded or showed interest in us asking and discussing this. How do you decide between biosimilar and Infliximab if given a choice?
Well, at the end of the day, I made the first step of the decision is really Infliximab versus an anti-TNF versus other mechanisms of action based on the patient's risk factors, disease severity, and how they're clinically doing. And then once I've decided on an anti-TNF, the second decision tree is what route of administration I'm going to use. And that's actually a discussion that we have with the patient. So if it's a matter of how do I decide between branded Infliximab or originator versus biosimilar 1, 2, or 3, at the end of the day, the counseling is about Infliximab. So in all honesty, the ultimate decision is not up to really us. It's about the insurers or the healthcare system. But most importantly, to reassure your patients, the decision about Infliximab is between the healthcare team, the patient, and any members of their care team.
Yeah, and I'll just further that and give you the practical answer that I've seen again in Cleveland. So there are 3 biosimilars for Remicade currently. The way that our health system works, we're using one of the biosimilars. So right now, for me to actually get biosimilar 2 or 3 Remicade, the second or the third that are out there, would be very difficult unless the patient's own individual insurance company actually mandated that biosimilar. So I am comfortable with the biosimilars that are out there. I'm comfortable with newly starting and single switching. So if an insurance company or a health system says this is what we're going to use in place of Remicade, and then soon to come Humira as biosimilar Humira becomes commercially available, I'm fine with that, and I have no problem with it. To the point of the question, it's probably not going to be up to the individual physician's choice, at least at this point. Tina, you brought up a question that now has a lot of likes and hits. Are biosimilars safe in pregnancy?
Mm-hmm. So the honest answer, do we have, does the piano registry have a subset for biosimilars? I'm not sure yet, but I'm sure that's something UMA is going to look towards. But I think that the same way that we extrapolated indications based on the clinical data for rheumatoid arthritis, ankylosing spondylitis, and also the multiple studies looking at originator versus biosimilar anti-TNFs, I actually feel pretty confident that we can extrapolate the safety data that we know about anti-TNFs during pregnancy and breastfeeding with the biosimilars as well, because we're using them in the same context. And remember, they're nearly identical. So they're like fingerprints to a certain degree. So the most important thing is you have to get mom in the best condition possible prior to family planning and conception.
Yeah, so I from a personal level, I've had pregnant women who've been on biosimilar Remicade. I think I'm fairly certain Uma's, I don't think on the line with us now to chime in, but I'm fairly certain in the piano registry, which is the registry of pregnancy, looking at different medications, that there were biosimilars, and to my knowledge, there was no issue or concern. Uma, can you speak to the use of biosimilar in pediatric patients? Do you have any concern, anything different that you do? What do you think about that?
So, I think the pediatric indications for Infliximab for Crohn's disease are there, but they don't have it for ulcerative colitis. But, we use the same, and, I'd also have to defer somewhat to my pediatric colleagues as well. But pediatric clinical trials and biologic data are oftentimes already extrapolated from adults. And so if we have reasonable, and we do, safety and efficacy, immunogenicity, PK/PD data for the biosimilars in the adult patient population, there's likely not going to be an appreciable difference within the pediatric patient populations. And I think we've seen that for the vast majority of our biologic mechanisms.
I agree. And so I would not be concerned with that either. Understanding at this meeting, quite a few of the topics were about lack of trials in pediatrics. Right. But with biosimilars, I think it's the same message, to use the word same, that we can use them in kids as well. There was a question that came in, interestingly, about the other ingredients. This actually comes up with the vaccine now, but we won't talk about the COVID-19 vaccine and kind of what it might be cut with or other ingredients. But Well, I'll ask you the question. I don't know if you see it as well, but the non-biologic ingredients and how these may differ from biosimilar to biosimilar, especially with respect to possible allergies. So, for example, sucrose, polysorbate 80, monobasic sodium phosphate, and then there are others as well in terms of different ingredients. This is an excellent question. It's a difficult question to answer, but I don't know, Tina, have you thought about this, or what are your thoughts on that?
Yeah, and I think that actually was some of the concern and controversy about biosimilars, especially for the use in Crohn's disease and ulcerative colitis initially, because even though the definition and the study showed that these are highly similar, the active compounds are pretty much identical. The differences are in these inactive, not thought to be clinically meaningful substrates. Now, in the clinical studies and in the overwhelming data, that has not translated to any differences in adverse effects. It has not translated to differences in persistence of therapy, discontinuation of therapy, infusion reactions, etc. However, it's going to be a case-by-case basis. If somebody did have a reaction, let's say, to originator adalimumab, I would take pause before giving them adalimumab biosimilar. But if somebody had an issue with, let's say, Infliximab biosimilar, it would not make me concerned about adalimumab biosimilar if it were in the right context.
Yeah, so far, and again, we've had now thousands of patients who we've had again, it's a single switch. I haven't seen this, but especially at first, I think it was more the nocebo effect that patients were worried. I did actually check antibodies to infliximab, maybe a little bit more frequency initially. And honestly, there were none, the level was good, and we continued with the same biosimilar. But could there be rare events that come out down the line? Sure. And I think we see this with anything, and it's hard to make a completely blanket statement. So thank you for that question. Yeah. Tina, again, the idea of switching back and forth, that seems to make a lot of our audience uncomfortable. This, idea that someday we might have 7 biosimilar Remicade, 7 or 8 biosimilar Humira, and kind of each time the insurance approves it or the health plan approves it, you get a different one. What are your thoughts on that? And again, I know the data are limited, but what are your thoughts on that?
Yeah, at the strictest, probably scientific evidence level, there's likely not going to be differences with the multiple switches. However, what I feel uncomfortable switching back and forth between reference and originator, if it were one switch between one biosimilar to the reference and back, and they had previous exposure, I'm not concerned about immunogenicity. I'm not necessarily concerned about loss of response. However, if we have 7 or 8 versions of Infliximab, and there is, I would want more interchangeability data to be present to go from 1 to 1 to 1 to 1, or I'd want more transparency from the insurance companies about making those switches beforehand so that we can keep track. Because I think this is really where pharmacovigilance is really going to be key to see how safe and effective these multiple switches across various biosimilars are going to be. Very comfortable with single switches. I think we need a little bit more data to truly meet the FDA definition of interchangeability.
I completely agree. And I think just to drive home the point, I think you said this earlier, is that there are some limited data on multiple switches, primarily in Europe, but that we have not seen any higher rates of antibodies, higher complications, but we just simply need more data. Tina, the last part of your talk was on communicating with the patient, and one of the questions that rose to the top was, how do you convince a reluctant patient to switch, especially if they're on their second or third biologic in general? So, you found something that works. It's an innovator reference biologic, we'll say Remicade, for example, or Humira. And now the insurance plan says, you know what, they have to switch. How do you make the patient more comfortable about this?
Yeah, and so it's not something that's going to happen over a 2-line, epic message. This is going to require a conversation over multiple parts. I think it's about just providing reassurance about with the data about with biosimilars and the reference product. Assure that you're going to do the same amount of monitoring that you would for the reference product, And for this type of circumstance, at the end of the day, we want to make sure that, and after the patient receives their first dose of the biosimilar, that you have close follow-up. So really, it's about communicating that they're not getting an inferior drug. And I would also say that try to address potential nocebo issues beforehand. And, in these particular circumstances, just really make sure that the switch is, if there's any issues, that it's not because of the switch. So for example, if somebody is switching from branded to a biosimilar, make sure that you have a calprotectin, make sure you have your drug monitoring or things like that beforehand, just so you know that if there's an issue, it's not because of the biosimilar, it's because of their disease state. So what I mean by that is, let's say you checked somebody's Infliximab level who had a rough course and a history of antibodies in the past, and their Infliximab levels were high, and then they got their biosimilar and they had an infusion reaction. Well, that's not necessarily due to the biosimilar. That's due to the fact that they had antibodies, or their calprotectin was starting to creep up and they flared after the biosimilar. Not necessarily due to the biosimilar. It's because their disease state had changed.
Yeah, and it's interesting now with telemedicine, telehealth. I'm doing a lot of this counseling in the last year. Obviously, we all have, but I find this is particularly good because I can reassure the patient. I can get on a telemedicine visit. It can be about 15 minutes. Like you said, it's not a one my chart message back to the patient. It's a 15-minute discussion. And I have had the times I've seen it are patients who've been, say, on, again, I'll use Remicade because of the biosimilar to Remicade, who almost had a colectomy years ago for severe UC or very bad fistula. And they've done extremely well. There's a lot of normal trepidation and angst about that. And I get it. It's hard for us to excuse that away or say, oh, don't worry about it. And in some of those patients, you said something important. Even when we're on our virtual visit or telemedicine, and I've reassured them, I say, you know what? Let's, knowing you're going to switch, let's check a calprotectin now. Let's check a level now, or let's get some objective data. And then let's do that again, maybe after a dose or two. And I'll say, knock on wood, fortunately, whatever state they were in, they're still in after the switch. And I think that's comforted them. So Tina, we have about 5 more minutes from questions, and I'm going to do kind of rapid fire now. And we'll kind of do quick questions, quick answers. Great to know drug monitoring. Is the dosing the same across the biosimilars for a branded drug?
Yes.
Okay, that's a really quick one. Have you had any situations in which a patient has had more than one switch, and do you have any concerns about switching drug inductions of the medication, during inductions of the medication?
And, I think we touched base upon this a little bit earlier. I'd say a couple of years ago, I may have, but at the end of the day, we have to treat the patient. And the most important thing is to get the right dose at the right interval, stay on schedule. So, and in terms of, have I had patients who've had multiple switches across different biosimilars? Not yet, but yeah. Is it coming? Yes.
Yeah, no, I'm the same way. We've had single switch so far. No, I shouldn't say that. There are a few patients who moved from somewhere else who are on Remicade biosimilar. They came to Cleveland, got a new job, got new insurance, and now they're on a different Remicade biosimilar. It's a small number, haven't seen a problem yet. There's a good question. This is kind of the part about transparency of marketplace and how do you handle this. So in certain regions, actually the insurers are not requiring biosimilar Infliximab. And that means that to them, to the patient at least, it's not cheaper, or the insurance. So what gives? Who benefits from the cheaper product anyway? And I think, Tina, we kind of talked about this. It doesn't go back to the provider. It doesn't go back to the patient. What do you think about that? I mean, I think that's a real world good question.
And so, where at the end of the day, let's be honest, it comes to the pricing, right? And how the pricing of the biologic is to your particular healthcare system, your academic institution. And is it, do they get a special pricing with reference versus a different pricing for biosimilars? So what I will say is, as we have more mechanisms of action available and more options. The biosimilar uptake is likely going to increase. So I'd be surprised if all insurers are going to stick to just the branded products in the future. So even if your region hasn't moved towards biosimilars yet, it likely will at some point. So at least being familiar with the nomenclature and the evidence actually will help you prepare for when that day comes.
Great. And then how about a question on, is there a pipeline for biosimilars for ustekinumab, vedolizumab?
I mean, almost certainly. Those aren't cheap either.
Yeah. So the answer is yes. It's just that Infliximab and adalimumab are the ones that we have more data on. A couple more questions and then we'll start wrapping it up. The PK/PD data and equivalency. We didn't really go into that too much. Maybe I'll just answer it. Based on the information we have from innovator reference, say Remicade, to biosimilar Remicade, the PK/PD data was the same. We didn't show that, but we have that from the Northwich and some other data as well. And Russ Cohen appropriately wrote, that biosimilars are not biosimilars to each other, only to the originator. So Russ, thanks for that comment. That's a very important point to point out. And I think I am going to now wrap it up. But Tina, again, outstanding job. I always enjoy doing these with you. The audience was very engaged. We're sorry we didn't get to all the questions, although I think we got to many of them and many of the themes. And then just to wrap up, I want to remind everybody to visit CMEOutfitters.com. Again, CMEOutfitters.com to access the presentation and other resources within GI. This is all accessible to you. Tina, again, I want to thank you for this great program. I also want to thank CME Outfitters for their assistance during this program and development of the program. As always, we enjoy working with them. I want everybody to stay safe, stay well, enjoy the rest of the conference, and remember to claim CME. Please complete your evaluations. And with that, thank you, everybody. Have a good night. We'll see you soon.
Thank you. Good night.